Good afternoon, everyone, and welcome to the second annual Ted Poyser Lecture in Soil Health. My name is Mario Tenuta. I am the Western Grains Research Foundation and Fertilizer Canada Senior Industrial Research Chair and for our Nutrient Stewardship and Professor in the Department of Soil Science here. I would like to acknowledge that the University of Manitoba campuses are located on the original lands of the Anishinabeg, the Ininawuk, Ansun-un-Nuwuk, the Dakota Ayate, and the Dene peoples. And of course, on the national homeland of the Red River Métis. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to moving forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. Before we welcome this year's Poyser Lecturer, I'd like to acknowledge my fellow members of the Poyser Lecture Committee. The chair, Mr. Tim Sopak, representing Manitoba Habitat Conservancy, and you're going to have the pleasure to hear some words about the lectureship from Tim in a moment. Laura Rance-Unger of Glacial Media. Laura, are you here? Probably joining us online. And Martin Entz, he's on my second page here, I have to go here. Martin Entz, the Jarolowski Chair in Natural Systems Agriculture for Climate Solutions and in the Department of Plant Science at the University of Manitoba. And also Crystal Jorgensen, Communications Coordinator of the Faculty of Agricultural and Food Sciences. Martin's back in that corner over there, and Crystal, I saw Crystal. Crystal's over there. Thanks all for making this Poyser lecture happen. I'd like to call now upon Mr. Tim Sopak to speak on behalf of the Manitoba Habitat Conservancy and provide some background on the Poyser lectureship. Tim has been instrumental in initiating the Poyser lectureship 2 years ago, and in organizing this year's lecture. He's really the chair and the one that instigates our meetings and sets them up and coordinates them, so that's very appreciated, Tim. He will now give us some background on the Poyser lecture. Tim?
Thank you, Mario. And on our steering committee, I'd also like to acknowledge Manitoba Habitat Conservancy. Samantha German has been providing our link back to the Conservancy. About 3 years ago, I retired from the Manitoba Habitat Conservancy. But along the way, I think the reason why I'm here is because I was a longtime friend and colleague of Ted Poyser. After a fulfilling life, Ted passed away in 2020 at the age of 93. And if you're wondering why, we are celebrating a guy named Ted Poyser who's long gone. Well, if you've ever used a Manitoba Soil Survey report, if you've ever taken advantage of the provincial crop insurance system, if you've ever benefited from a project with the local watershed conservation district, or taken advantage of any number of soil, water, and wildlife conservation projects in this province and beyond, delivered by a number of organizations, then you should know that Ted Poyser spent his life helping to make those things happen. He was born on a farm in the McGregor area in 1926, grew up through the 30s, the dirty 30s, and he was stimulated to take a degree in agriculture, followed up by a master's degree in soil science. And he took that degree under Dr. Joe Ellis, whose name now is on the soil science building here at U of M. Ted could easily have pursued an academic career. He's a very smart guy, but he literally started at the ground level, working in the provincial soil survey. He then worked on a number of soil conservation programs. He was instrumental in establishing Manitoba's first watershed district. And from there, he worked his way up to leadership positions in major agricultural land use and land adjustment programs in the province and beyond. You know, for example, he played a significant role in the establishment of the crop insurance program. He led the Canada Land Inventory in Manitoba. He led a major agriculture and rural development initiative in the Interlake called Funds for Rural Economic Development, which is really a groundbreaking initiative for land use and rural development. Land assembly for some of the biggest provincial parks in southern Manitoba and some of our biggest wildlife management areas occurred under programs that he led. But through all that, really what he cared about the most was people. It started with farmers sustaining their livelihoods on the land in which they worked, but it also included rural community dwellers and on up to urban citizens who might be looking for a chance to be out in nature. As his reputation grew, researchers, middle managers, senior managers, and cabinet ministers of all political stripes sought out Ted's views. Among other things, his key message was that when it comes to the land, conservation starts with the soil. While most of his career accomplishments occurred in the 60s, 70s, and 80s, so a lot of you in the room would have no connection with that. His behind-the-scenes influence carried on much later than that. So well into his 8th decade of life, he was being sought out for his views. And one of the reasons was that he excelled at helping people to see the world more broadly. He was a holistic thinker in the world of soil, water, and wildlife conservation. He could move seamlessly through those areas where people saw stovepipes, he saw opportunities. Many people who became leaders in agricultural research, government policy planning, program delivery, will tell you that Ted was one of their most important mentors, and I would certainly add my name to that. After he died, the Manitoba Habitat Conservancy, Ted's family, watershed districts, the Manitoba Soil Science Society, and many of his colleagues contributed to the fund that supports this lecture, and we call it the Ted Poyser Plant Ideas Fund. It's an endowment that's held at the Winnipeg Foundation. The name expresses really what Ted did best, which was stimulating people to think in new ways. It's fitting that the Conservancy manages this fund because Ted played a foundational role in helping it to become Manitoba's largest habitat conservation organization. I'm also tremendously gratified that we have the Department of Soil Science and the Faculty of Agriculture who have embraced the annual Ted Poyser Lecture in Soil Health. And once again, just to echo Mario's thoughts, thank you so much to the steering committee for all the work behind the scenes, and to the main attraction. Laura, thank you.
Thank you, Tim. Before we begin the lecture, here are some instructions on how our online audience can ask questions during the Q&A session following the lecture. If you're watching on the Sustainability of Canadian Agriculture Conference platform, just to enter your questions into the Q&A panel that you will see on your screen. Now, if you're watching us today through YouTube, please open a second screen on your browser and visit www.slido.com and use the access code number, it's more than a number, SCAC2025, so SCAC 2025, okay? All together, no spaces. These instructions are also in the YouTube description. I'm not sure what that refers to in the YouTube description, but I hope you can find that out. But anyway, use SCAC 2025. At the end of Dr. Van Eerd's talk, we'll take questions from both the in-person, that means you folks here, and for those in monitor land. And now I'd like to introduce you to the 2005 Ted Poyser Lecturer in Soil Health, Dr. Laura Van Eerd. Dr. Laura Van Eerd is Professor of Sustainable Soil Management at the University of Guelph Ridgetown campus in Ontario, specializing in soil health and nutrient cycling. Her internationally recognized research helps farmers improve soil care, crop productivity, and profitability. Her innovation and foresight led to the establishment of a state-of-the-art long-term experiment at Ridgetown that has advanced global knowledge on cover cropping in temperate climates, and I'm sure we're going to hear much about the great findings and results from that long-term study. A highly cited author, she has received numerous awards for her contributions to research, teaching, and agricultural leadership. Dr. Van Eerd co-led the Soil Health chapter in the Canadian Society of Soil Science textbook, Digging into Canadian Soils, an Introduction to Soil Science. Laura has received many awards for contributing to teaching, research, and extension. She is a fellow of the Canadian Society of Agronomy, recognized as a soil champion by the Ontario Soil and Crop Improvement Association, and included among the most influential women of Canadian agriculture. Those are some pretty awesome recognitions there, Laura. Please join me now in welcoming Dr. Van Eerd as the 2005 Ted Poyser Lecturer in Soil Health.
Wonderful. Thanks you so much. Right. So I also want to start with a land acknowledgement of the place where I live and work at Ridgetown. And here's how our university positions this acknowledgement. I'd like to add my personal acknowledgement that the lands of a dish with one spoon have been cared and managed for by generations in memoria. My learning about truth and reconciliation has been in various forms. Two I'd like to acknowledge right now. One is with the stonefishes who have shared their food, their community, and a conversation about what truth and reconciliation might mean to an individual. And I truly value that. The second is my interactions and relationship with Melissa Arcan, who does the same and then positions it relative to soil and agriculture. And that has been super valuable for me. She publishes on it so that all of us can learn. And I am very grateful to follow Melissa in this lecture, to be the second presenter here. Miigwech. So where's Ridgetown? We are 10 minutes from Lake Erie. The Great Lakes modulate our climate, so less extreme in the winter and summer. That influences our production system. This is important because context matters in terms of agronomy and soil science. And so I'm really happy to share what we are seeing in Ontario and how that might apply in your system, in your soils. So we get about 80 millimeters of rainfall monthly on average. However, in July and August, that 80 millimeters isn't enough to overcome evapotranspiration. And so we do see soil moisture deficits in our climate. So, Mario, you described my research quite appropriately. I guess I would add that I'm kind of at the intersection of soil science and agronomy, kind of what the plants do to the soil and how the soil affects the plants. It's a nice synopsis. Most of this research has been in nitrogen use efficiency, cover crops, and then soil health. And so that's the context from where I'm coming from. What do we want to do today? I would really like to define how I see soil health. It's not the only way, but it's the context with which I come from in this talk and the research that I do. We're going to use the long-term experiments to kind of make this link between soil health and productivity, looking specifically at cover crops, crop rotation, and tillage. And then we're going to go to see what's happening on farmer fields. What are they seeing? What's being done in Ontario? How does that work? And then in terms of future research needs, I'm going to hit that right now here. What I see, so it's kind of the boundaries of what we're finding, is that if you have a soil health test on your farm, what's the recommended outcome? What's the recommended action that you take based on that score? And we're not there. We will never, in my personal opinion, will never be like pH, therefore apply lime, or therefore do this. But more research would be much appreciated. And then today I'm going to scope it as future research, that link between yield and resiliency on the farm and how that works. Yeah, right now we're collecting samples on the farm, but we don't have it linked to yield, right? And part of that is social in terms of asking a farmer to provide that data. So we'll leave it there in terms of future needs. All right, so how do we define soil? Healthy soil is one that functions, one that works. It's hard for me to believe that it's been a decade since World Soils Day. I still like this diagram. It shows the multi-functions that we expect from a healthy soil. And we can link these functions to an indicator, a test that we measure, maybe in the lab, maybe in the field, of is soil doing those functions? So we can measure the soil health test, we can measure directly these outcomes. And so that's how, when I say thinking about soil health, I'm thinking about what the soil does. Linking that to resiliency from this agricultural perspective, I think of a resilient system as one that is less dependent on inputs, can produce crop productivity without a lot of inputs. And I really like this schematic where the puzzle pieces are the management. What are the managements that you can do on your farm? And when you link them all together, hopefully you're less reliant on inputs. I guess kind of the add-on to this is, does it increase yield? And that's kind of the question mark. Okay, so then what do we do? What can farmers do on their fields to enhance soil health? 6 C's, I gotta be careful because if I say it really quickly or if I'm really nervous, it sounds like something else, but it's equally sexy. So what are the 6 C's? They're carbon, and they all start with C, so it's compost and amendments, compaction reduction, cover crops, complexity, and we'll include crop and animal, continuous living plants, and conservation tillage. They're the known best management practices. Well, you can call them sexy if you like. They're standard, and I'm going to explore the cover crops here. It's not short term, it's long term, right? So, and then so how do we pull this all together? The function, the indicators, this schematic from the textbook that you mentioned, Mario, right? Do you have a threat to your soil? How are those measurements? So those are the soil health indicators. Take action and then hopefully observe those outcomes. I guess I would add that it's a continuum, yes, but you can join into this matrix at any point. Maybe you're seeking more from your soil. And so you're like, well, I would like more of this observed outcome of nutrient supply from my soil. So moving backwards to say, okay, here are the actions that I'd like to take to get this outcome. All right. Straight lines, maybe not, but it's a good conceptual model. All right, so we've defined soil. Where have we measured it? Well, in long-term trials. So I'm going to take a moment to describe my long-term cover crop trial. Here we have 5 different cover crop treatments, and they've been in the same area, that main plot of 16 meters by 6 meters wide. It's in a horticultural system, so we grow processing vegetable and then grains and oil seeds in this rotation. So in southern Ontario, we still have processing vegetable, and it fits with the soil type. So if you would come to this experiment in the summer, you will see that the main crop looks quite homogeneous. Where the treatments are applied are in the fall after main crop harvest. We do tillage to prepare for the cover crops. We do tillage to prepare for the main crops. So we're setting up the crop for success. So in the background, you can see the corn. That's the 3 reps. There's actually 4 of corn that was planted in 2020. That's site A. In the foreground are the cover crops, and they were planted after processing tomatoes. That's the site that's a year behind. So we established where the corn is growing. It was established in 07. A year later, in 08, we established the same 5 treatments, different randomization, same order. Okay, so now we've got some added power of extra plots, but also that year-to-year variability. We're an orthohumic glacial. It's a luvasol that's got some drainage. So I think it's fair to classify it as a glacial. So nice sandy loam texture. Organic matter is quite good. So this is nice forgiving soil. pH does range. You can see our Olsen P and K values. So pretty representative soil type. Again, we're in southern Ontario, so we can grow some nice cover crops. And because we're in a horticultural system where our vegetables are harvested early, we can plant and grow cover crops for a longer season, right? So this is one of the contributing factors of what we're seeing in our system is long growing season and high frequency of cover crops in the rotation. So this is oats, and you can see the planting dates there. So they're in site B. The other site is a year ahead. Okay, so in the medium term, so our early, let's start with the early term, right? First year cover cropping. We were looking at nitrogen dynamics and how nitrogen contributed to the next crop. In the medium term, so here after 8 years of having the experiment going, in that 8 years, we were able to plant cover crops 6 times. So high frequency because of the number of processing vegetables in there. Okay, so what did we find? Shifts in the microbial community with Dr. Carrie Dunfield. We found increase in activity and tomato yields. And when you add the economics of tomatoes, then we get quite economical to include cover crops in the rotation. And then this is now Dr. Inarjot Chahal's work, and she looked at soil health indicators. So what was available a decade ago were the commercial indicators, and we put them to the test. So after these 8 years, could we see any differences in these commercial soil health tests? And we did see that we could pick up some treatment difference. It was not always the same with every cover crop, and it was not always the same with all the indicators. Overall, in our system, we could suggest that the Haney soil test is less able to pick out treatment differences than the other ones. The other ones seem good to look at, I guess, in further explorations. Right, and all of this becomes part of what might work in Ontario, what might work in this system. So that was in the medium term. And then in 2019, the Soil Health Institute was doing their North American-wide survey, and they soil sampled. So now we have an independent quantification of soil health in our system. And yeah, so we did see that some indicators, up to 15% increase in soil health. Not a lot of those indicators were significant. Only about 5 out of the handful that they did. However, we were showing the similar trends of what we had seen in our earlier medium-term study, which was enhanced soil health. Part of the limitation of just looking at our site with the data set is there's only N of 16. So they only sampled 16 plots because their goal is to look North American wide. So with a smaller number of samples, we just couldn't pick out those differences. Whereas in the medium term, right, our number of observations was 40 in each site. So now we've got 80. Now our power to detect differences has increased. So that's a little bit of foreshadowing about when we scale to the farmer's field, right? And the amount of sampling and how do we quantify the diversity at the farmer field level. Fair enough. So we see some of these changes in the soil. How do the plants respond? So in the spring, the Soil Health Institute comes to measure soil health, and then we grow tomatoes. And at this time, we collaborated with plant pathologists to look at tomato disease. And so that's the scouting over the season, looking at the crop and then looking at the fruit. And what we saw in both years was in the no cover crops, the tomato plants senesced quicker than with the cover crop. So those plants just said they shut down, and so it's kind of like which is the chicken or the egg. The soil scientist says, yeah, the soil is influencing the plant, and therefore the plants are shutting down. I'm not saying that. That's all I'll say there. So they shut down quicker. This then, with the lack of canopy, what would come in would be disease, right? And so we have that definite chicken and egg. And we did quantify statistical differences in anthracnose on the fruit. So in 2019, year conducive to anthracnose pressure, we see quite a bit of decline with long-term cover cropping than without. And these are at the scale of what we might expect if we have a fungicide trial, those experiments. We also had a 0 nitrogen to the corn versus full nitrogen to the tomatoes. We had 0 nitrogen to the tomatoes or full to the tomatoes, and we saw the nitrogen impact on disease, but it was less than the long-term cover crop impact. So this was in 19, and then similar but not as high disease pressure in 20. When you consider that even one lesion on the fruit impacts grower yield and their amount that they're paid, then the economics for cover crops are quite simple and easy to pencil out. So now we see this interaction. I'm going to put myself out there and say this is resiliency, right? The ability of a plant to withstand disease pressure. We follow the rotation. After tomatoes, we plant cover crops, then we plant corn. Here, now we've got 4-year data. We have each bar is a number of observations is 32. And we see this yield response to long-term cover cropping with corn. Now we push the system. We didn't apply commercial fertilizer to the corn, right? Because we want to see what's the value from the cover crop. I can tell you that county average yields are around 12 ton. And that's commercially. So one, we're starting with nice productive soil, and we've shown an impact of long-term cover cropping. And it's not an excessive amount of time where we're seeing this crop response. So now let's relate that to the Soil Health Institute's number. So we have the graph of soil organic matter at the top, where not statistical difference, but our crop is responding to it. So this is their numbers of soil organic matter plotted against 2020, our most recent yield. And we see this quite strong relationship between soil organic matter and grain corn yield. Now, 2020 happened to be a drier yield. And so we asked, is it the nitrogen? Is it the water? Is it the water and nitrogen? That's, if we go back to the function that we expect from a cover crop. And so we set up experiments to test this. And so the first thing we did, so this is Yajin Peng, who is now a postdoc with me, but she did her PhD and she quantified soil organic carbon in our experiments. We had what the Soil Health Institute did, and now she broadened it and went deeper through the profile. And we see that we are increasing soil organic carbon. A little bit surprised that by depth we're seeing that treatment difference. Yeah, so, but we have pushed soil organic stocks. So this is on our main plots with cover crops. Now we have large enough plots that we could ask the question, is it the long-term cover crop, or is it just because we had cover crop? Is it one time versus no cover crop? And so we set up this experiment where we sacrificed a small amount of a quarter of our no cover crop control, and we planted cover crops for the first time. All right, so you can see that there. And so our hypothesis, well, what would you hypothesize? Is it the cover crops long-term, or is it first time? Yeah. So we've had another graduate student, Polly, who followed nitrogen dynamics in the crop and in the soil throughout the growing season. We could see deficiency in the no cover crop. The first time was intermediary. This was true in the green seeker and SPAD meter. This was true in any of our measurements. Which one do you think had more soil moisture? The smaller plant, right? So it didn't line up as how we had thought, right? We thought there'd be more moisture because of organic matter and the provisioning of water, but it was just more corn growth and more evapotranspiration. Yield. Oh, so you can see how new this data is because it's still in bushels per acre. Yeah. Again, intermediary. So there is some there is some effect with first time, absolutely. Not enough to say statistically different from either of them. But we're seeing that it is the long-term crops that are contributing to what I'll call resiliency. And then Yajin kind of was like, well, where is this nitrogen coming from? Or where is this source. And so she did some path analysis, she did some fragmentation, and in this path analysis, she chose the palm, so particulate organic matter that was sampled in the spring, and then tried to relate that to grain corn and grain nitrogen uptake. And so what her findings are showing is that it's the particulate organic matter that's driving the yield and the nitrogen uptake. I guess not surprising, because this was done solely by fractionation, and not surprising because we're on a sandy soil. So lots of palm there to begin with. Really curious, our next step, and Yajin really wants to look at, what's the mineral component and break it up into smaller fractions to see how that's contributing to the system. Okay, so take home, we have healthy soils. They have many pathways by which the crop can uptake nitrogen, right? It's more pathways, less deficiencies with healthy soil. So that's our long-term experiment on cover crops. We've gone to other other long-term experiments where we've looked at tillage. And here's our soil health scores from 4 different long-term experiments in Ontario. And we see the advantage of no-till to soil health. Again, that's about protecting the soil compared to plow. And then we have 2 long-term experiments at the University of Guelph, at Elora and at Richtown. And here we did a synopsis of crop diversity. In our temperate climate, it's the inclusion of winter wheat into a corn-soybean rotation that is really helpful in terms of yield, but also soil health. And red clover to some extent, alfalfa definitely, but that winter wheat alone is a huge contribution. And so we gathered up, we being Cameron Ogilvie with Soils at Guelph, led fact sheets. So we have 4 fact sheets that summarize the findings from our long-term tillage and crop rotation experiments. And these fact sheets summarize the papers that are shown here. And the bottom line, with winter wheat in our system, we have healthier soils, greater yields, more resilient. That is better yields in those off-weather years. So too wet or too dry. We're talking 2 to 5 bushel of corn more with having winter wheat in the rotation, up to 20 bushels in soybeans with having winter wheat in the rotation, and less reliant on nitrogen fertilizer with winter wheat in the rotation over the long term. Okay, so that's our Ontario data. It scales to our long-term experiments in North America. They'll recommend this paper here that shows crop diversity. And it also scales to the Soil Health Institute's finding about cover crops and crop diversity. So our long-term experiments, I think we have quite a bit of evidence to support that. Now, can we take it to the farm? Here we go. So I got an opportunity to work with ecological farmers, and Sarah and those farmer-led research said, can I measure soil health on my farm? And what are the best indicators? And can I compare a good field to a poor field? And does it work? Does it match with expectations of productivity? And so Sarah designed this, so quite intensive sampling. So here's the key. It's not the precise way of of doing it, but it's that you have many samples, many replication to account for the diversity in your fields. The indicators available at that time and that were of interest to farmers are shown here. Samples were sent to the states for analysis. So let's see, what were the outcomes? Well, there's two indicators that could distinguish between the high productive fields and the low productive fields. One of them was active carbon, so that could distinguish between grower identified fields. They were consistent across these different farms. So those farms were hay, vegetable, or rotationally grazed pastures, so much different systems. So it's organic carbon came out of this analysis, and soil organic matter was also reproducible as long as you had a half percent difference you could pick out. And so that was based on power analysis. So excellent opportunity, intensively sampled with grower knowledge of where to sample as well. You'll see here we had a reference site. That reference was a less disturbed fence row or something similar that the farmer had identified. What else is happening on the landscape in Ontario? Well, the Green Belt, which is around Toronto, the counties around Toronto, the Green Belt, they're looking at testing soils, farmer fields. They are sampling a reference. They're sampling soils or fields that have a history of winter wheat and cover crops and less tillage. So they have kind of these three paired sampling areas. And then the indicators, those indicators are from the Soil Health Institute. They are combustion for carbon. The carbon mineralization is a 24-hour burst, and the aggregate stability is probably more accurately called a slake test, 10-minute slake test, where you put an aggregate on to look at that. And then we've included our SHAP, which is Ontario's SHAP. So what's SHAP? SHAP was developed by the Ontario Ministry of Ag. It's a framework, and it's a tool that we had anticipated CCAs might use. We'll leave it there. So we kind of, the whole thing is a better understanding of soil, and ultimately that bottom part, which is kind of what we had talked about at the beginning in terms of, future research. Future research was the action that you take. And so this framework is working towards taking action based on your soil health score. Okay, so SHAP, here are the indicators. So this was done by OMAFA based on the literature available at the time, based on Ontario and other regions, based on expertise. We are looking into these indicators. They're not set in stone, but they're the starting point for what we're doing. So then if we look at the SHAP tool, it's trying to not just say, here's your lab test. It's integrating it with your inherent properties, your management. And then you have add-on modules of what are you seeing in the field? What's your other risk? And then your management index. So pretty comprehensive. lots of ways to come into it. Yeah, and in the end, you get a report from the analytical part, and we hope to get towards solutions and a monitoring plan, which we see there in blue. Okay, so if we're gonna have a SHAP, then we're gonna generate a report, we need to scale it to the rest of the province. And so this is, we had a photo of Sebastian there. Now we have Dan Sarret, who has provided leadership on the topsoil program, where went to farmer fields, government knocking on the door, can we dig a hole in your field, take some and put it back in. Oh, and I'm from the government, I'm here to help. Yes, that is not my words. That was someone who is involved in the program, not the person pictured here. Okay, so you go to the field, you ask the farmer the questions. What type of management are you doing? Record that, you collect a sample. And quite fortunately, they shared this initiative with the University of Guelph researchers so that we could add our special soil analysis to these soils. Okay, and you measure those indicators. And with this 1,500 data set, we could build scoring functions, right? Just a normalized curve to get to your scoring functions. So if your organic carbon lies at this rate, you go up and you see which color you land in your score. We tested if we would need 5 categories of soil texture, because we know soil texture influences carbon and other soil health indicators. So we tested, do we need 5? We never needed 5 to show differences in our scoring functions. Some indicators you need 3 different soil textures, some 2, some 1. So if you want to know more, the Topsoil Project report is available online. You can access it there, and Interjot's publication is cited. Okay, so now we have scoring functions. Now we can say how this field compares to the rest of that 1,500 samples in Ontario. Interjot's interested in those relationships. So does those management, so tillage intensity and cover crops, organic amendments, how do they relate? So some principal component analysis there. It's the system. It's the system. Anytime we're there, what you grow and the management of that system is the main outcome. So here's the breakdown of the cropping system. So you can see there, Ontario is mainly a grain production, and so most of our samples are from grain system. And you can see the texture there, the breakdown. I guess we need to build, if we're going to have a SHAP test, we need to build. Oh no, are you okay? Oh, we need. I know it's okay. Okay. Sorry. We're going to build the database. And the reason why is because if you have a number of samples of 33 in an orchard systems, we don't want to make inferences from that system with such a low number. It is representative of the province though, right? We have less orchards in Ontario than grain in terms of acreage, but yeah, we're looking to build the database. And so we have a ground truthing SHAP project to add 1,000 samples to the database. And instead of the topsoil program where they went out to sample, right? Pedologists went to the farmer field to sample. Now we're asking farmers, CCAs, and our partners to take the sample and to, right? And the reason to do that is to build capacity. And as instead of a push, it's more of a pull on soil health. And it is a leap of faith, right? So in addition to those soil health indicators that are part of SHAP, we offered through this program to do organic carbon, total nitrogen, and then our fertility package. So the typical fertility. We can offer one sample per field that we can pay for. We ask that growers provide their management on these 6 principles of soil health, right? Compaction, reduction, organic amendments, cover crops, residue removal, tillage practice, and cover crop. And so all of this is done through an app with the drop-down menus and clicks. So relatively easy, but you need to know the farmer field and you need to agree to provide that data. And that data is secure and anonymized. And yeah, it's still data, and we need the point location because of the soil characteristics and soil mapping. So here's what we found so far. Again, of all of those 6 things, it's cropping system that comes out. And texture, but texture we're going to deal with because we have the scoring functions that are based on texture. And then we had an awesome opportunity to collaborate with Cornell, combining the greenbelt data, the topsoil data, our ground truthing data, because we have the management, to look at the EU's soil degradation index. And there, they want to look at SOC, so soil organic carbon to clay content. This ratio shows potential for degradation. Very much skewed by your inherent properties. And so those lines across the box plot kind of shows if you're at the low end, you would be classified as a degraded soil. And if you're up here, then you're very good. So this is very heavily biased, right? And so we tested it on our data set. And we proposed a new method of looking at the carbon in pastures. So pastures less disturbed, known for carbon. And so here's our data set from New York and Ontario. We have a clay content versus how much carbon is stored. And then we use that ratio to put out a different scoring system. So this is SOC versus what would be expected at that clay content. So SOC versus SOC expected. And now we're being more fair to the soil texture that's there. Okay, so we have better lines, better scoring. I guess, how does that relate to other indicators of soil health? And so we chose active carbon, or POC, to compare these different categories of degradation. And so you see the SOC to clay, not much difference, not explaining a lot. But once we modify the ratio to what we would expect from a pasture, then we see this nice relationship. And so if you're in Ontario, we got concert tickets for you. Let us pay for your soil analysis. Yep, we'll cover the cost to build that 1,000 database. And the reason why is because we need Ontario data to have Ontario scoring. Yeah, it's been an interesting adventure with soil health testing. I'd like to thank my team. Really, really appreciative of all of their work and perspectives and knowledge, and I grow every day. And finally, the funders. Thank you.
First of all, thank you, Laura, for a great talk. And I apologize for the disruption there.
No worries.
I thought I was trapped in the room for a second.
I was worried.
Okay, so now we have the opportunity to take time for questions from our in-person audience and our online audience. Okay, and so for those of you watching as part of the, oh no, that's the next thing there. So we're going to start off open for questions. In-house, please raise your hand, or sorry, just come over to the center here to the microphone. And also Crystal's going to help with navigating online, any questions posed there. Do you have something online now?
Yeah, why don't we kick it off with one of the online questions?
Sure.
We have a couple here. And I'd like to just note that we had, at one point, we had 150 people watching online. So we've got a really healthy online audience as well. The first question that came in was from Ross McDonald. He asked, in general, soil health seems to ignore using native grassland soils as a benchmark. Do you think that they should be more fully recognized and examined as baselines for soil structure and microbial biodiversity in annual cropping systems?
Amazing question, and a little bit was answered with the pasture. I wouldn't say benchmarking. I think so. There's a couple of things there. There's the purpose of the benchmark. I think anytime you look at different soils, you're gaining in knowledge. You're gaining in what does it look like? How does it crumble? Right. So there's value in there. The value in testing, right? It's a nice comparison that ecological farmers research. They use that as a reference. We use this reference in comparing degraded index. So yes, the caution I want to say is that it's not a reference to say this is what our production system should be. And if you're not there, then you're horrible, right? It is an opportunity to learn about soil and to compare and say, yeah, we're different. But to say we want all soils to be like this, no, they're functioning. We want soils to be functioning. And so I'll answer it that way. So is that a good agronomist question? It depends.
Any other questions online there?
Yeah.
Crystal?
While the rest of you are thinking of really good questions, this one comes from Roland. Soil health currently appears to be derived through parameters that come entirely from lab analysis. Can you foresee a soil health assessment, possibly less accurate, that is based on readily observable or measure in place parameters, aka a DIY method for farmers?
Yep, thank you. So, yeah, and I think that's maybe a symptom that we're scientists and we like to measure things, and we tend to like to measure things in the lab. So the SHAP soil health test does have in-field measurements, right? And those in-field measurements are good. They can be very accurate, right? So one example would be an infiltration test. Set up, pound in a ring, measure how quickly the water infiltrates. That's a good indication of ponding and water holding and crusting even. So I think there's place for both. I definitely would suggest a shovel, and looking at soil is a very powerful tool. And for me, I'm not so much hung up on which indicator or that indicator or shipping it to the lab. To me, the value in this soil health is that we're getting people interested in soil, and we are getting people to look at soil. And that, I think, is the value of soil health and soil health testing.
While Martin gets to the microphone there. Now you've partaken in lots of research projects now. The soil health initiatives and the SHAP and your concert ticket there. In general, what's your sense on the reaction from farmers to the whole initiative? Is this something that they're all for? Is it something that they think is just a government ploy? What's your sense in terms of, is it more embraced than other replicated field plot studies and stuff like that?
Okay, so this is what I didn't show because I wasn't brave enough, but I see myself here in a cover crop field, and it's sunflowers blooming and the sky is shining, and we get to offer farmers to cover the cost of it. And then there's the photo of the shed in my town that's offering free hugs, and I often wonder if that's how farmers view what what it's like. And I think for many reasons, the one is I can only offer one test per field because I want to build the database across the province. So what do you do with one field? Well, I mean, okay, so you benchmark. That's what I think it is. It's a benchmark, and you're the best person because you know your field, you know the history, you know what spot to choose versus another spot. So we can offer a benchmark. I also offered a fertility test, but we don't fertility test in that manner, right? You would never benchmark for fertility. And so then, there's that hesitation. So right now we're at 40%, right? We had hoped to be at 65% of our 1,000 samples. So it's kind of that push and pull, right? Yeah. And I think that's just a symptom of where we are in terms of soil health and as an industry. And if I look back when I first started doing cover crop work, it was, yep, Laura, yep, cover crops. And now it's like, yeah, Laura, how do we do that? What do we do? And then so it will take time.
Okay, thanks very much, Laura. That was really good. So I've followed the long-term studies at the University of Guelph for my whole career, and I've really found them fascinating because I remember Tony Vine started it, and I read an article that talked about how the cropping system, as you said, was the most important thing to soil health. This was back along late 80s, early 90s. And no-till was also helpful, but they were sort of complementary. But the plant diversity was really important. So what I found fascinating is that you've got the corn soybean rotation, and as you mentioned today, then you layer the winter wheat on that, and that has this transformative effect. If you're nodding to that, and you mentioned that, and then if you add the red clover cover crop to it, it has even more transformative effect. It's kind of relevant to us because we're in an area where we grow corn, we grow soybeans, we grow a lot of canola, and we also grow wheat. And I'm wondering what thoughts you might have for this part of the world where we have that kind of rotation. And in particular, what is it about that winter wheat that, stimulates that system to better soil health? Thanks.
Yeah, great. So I would like to answer that question over dinner, Martin. So, yeah, what is it about winter wheat? So I'll just talk about the low-hanging fruit about it. I think it's temporally is different in terms of when root exudates are coming out into the soil and then therefore stimulating the soil biota and that's influence on soil functioning. I think that's the number one influence. You asked another part. What was the first part, really quickly, that you asked about?
Just what might be the lesson for
Oh.
our type of rotations here, where we kind of grow all those crops, but also canola.
Yeah. So I guess if I was here and knew, I would be setting up a long-term experiment to test that. The inclusion of soybeans, I think, is probably problematic in terms of soil health, just because of the lack of residue, shorter growing season, right? Yeah. So what else? I think, yeah, it's a question that I've been trying to answer, and I was hoping to go to the Soil Health Institute's database of their long-term trials. The problem is that they're dominated by corn and soybean rotations, and so we can't tease apart that crop diversity component because the only other one is winter wheat, right? So I would love to have an experiment where you have lots of different diversity and you could say, well, is it winter wheat or is it a 3-crop system? In my, my sense is that in our temperate climate, it's winter wheat. Yeah.
Thank you, Laura, for your wonderful presentation. So I had just curious about how would the long-term cover cropping would change the soil in organic carbon, bulk density, and soil infiltration. Yeah, I'm not sure if you have investigated these indicators.
So has changed, sorry, the last part, how?
Infiltrability, infiltration.
Yeah, soil infiltration. So the Soil Health Institute did do Centura, like the system to look at infiltration, and not a lot of differences. We're just really on that nice sandy loam soil that we're not picking out any differences in infiltration. And we have done some work with Dr. Richard Heck to look at CT scans and pore sizes. We see differences in terms of crust when we sample in like corn harvest in an undisturbed soil, right? We haven't tilled that spring. So we have looked at that, and we do see some treatment differences, in terms of void space. I guess you'd have to be because it's such a sandy loam soil, you'd have to be like saturated to begin with, and then try to look at infiltration, like that timing and water at the time of sampling really didn't allow for treatment differences there.
We have time for 30 seconds left for questions. So, Laura, you mentioned that Interjot is interested in the mineral-associated fraction. So that's on the next horizon. What else is on the horizon for your long-term study site? Because several of us have long-term sites here, and we're always either reinventing them, but you don't want to reinvent if you want to keep treatments the same, but we're reinventing our investigation, right?
Yeah, thank you, Mario. So with the long-term experiment, Jin is definitely interested in that mineral component and whether, kind of exploring more what is the contribution of those cover crops to those different fractions. We will this spring set out rain out shelters. So if any of, well, I guess if you've ever have experience in rain out shelters, please let me know. My guess is it's the quickest way to get a rainy season is to put out a shelter. So yeah, like kind of it goes back to, is it the nitrogen or is it the water that we're seeing in the first time, medium and long term? And one way to parse that out, I am hoping, is through those rain out shelters and looking at drought tolerance in the corn. We'll see. There's challenges to it, but we're excited about kind of teasing that apart.
Yeah, that is very exciting. And I'm sure your field technicians are really excited about having to take care of the rain shelters. With that, I'd like you to please join me and thank Dr. Laura Van Eerd for being this year's Ted Poyser Lecturer in Soil Health and sharing your experience with your research studies, but also in soil health, which in Manitoba, we're not that far along at all, actually. We're at the very beginning. We're like Ontario 15 years ago in past. So we thank you for your experience and sharing your ideas with that. Please thank you. So at this time, I have some instructions. For those of you watching as part of the Sustainability Conference, please be sure to come back to the platform that you're viewing at 1:30 p.m. for the first graduate student video session at 1:30 p.m. With that, thank you everyone, and have a good